makes the transition into organisms. So how why the more complex life has evolved over time. Back in 1995, Maynard Smith and Zaharescu proposed that this can be achieved through a series of major evolutionary transitions, including the origins of cells, genomes, genetic code, eukaryotes, sex, multicellularity, social groups such as social insects. Among those transitions, the origin of multicellularity allows opportunities for further complexity to evolve and led to more complex life we see today, like animals and plants. Across the tree of life on Earth, multicellularity has originated independently a number of times. The thing we want to know is that what are the minimum necessary steps that are required at first. Usually people do this by comparing those multicellular lineages with their unicellular relatives. But the problem is that those things happened a long time ago and many changes have occurred in the extant species, and which makes it difficult to infer what actually happens in the beginning. So one of those multicellular lineages is the green algae, which includes volvocine algae. This group of algae contains species with different levels of complexity, from unicellular chlamydomonas to fully differentiated volvox. So with this unicellular algae on top, we can replay life tape to see how multicellularity originates in the first place. The model species of the unicellular algae is the Chlamydomonas reinhardtii. It's a model system for photosynthesis and motility. Their vegetative cells or asexual cells are haploid, and their genome has been sequenced and annotated, which makes them a great system to do evolution experiment with, so that we can see how their phenotypes and genotypes change in real time. Among the possible selective pressures that might generate multicellularity, predation has long been hypothesized as a major cause for multicellularity because it favors bigger size, such as groups of cells over single cells. So here's a picture of ciliates and algae. Those feeder-feeding ciliates, they are also unicellular. They can feed on those single-cell algae. However, if we continuously expose those algae to ciliates, that will generate the selective pressure to make them become bigger or form multicellular structures. So a few years ago, my advisor, Matt, and Jacob, a graduate student in our lab, did this evolution experiment to select for multicellularity using predators. So the experiment was started with an outcross population by mating two different algae strains. So we have genetic diversity in the beginning. The starting population was split into 5 experimental populations that were cultured with predators and 3 control populations that were cultured without predators. Those cultures were transferred into fresh media every week over a year. By the end of the experiment, with 2 of the experimental populations, B2 and B5 population, they evolved those multicellular structures not observed in any of the control populations. So here's a time-lapse video over 72 hours of a unicellular algae strain. So you can see that when cells go through cell division, there are some little clumps there, but soon after that, the daughter cells will separate upon cell division. Here's one of our evolved multicellular strain. And you can see that the cells stay together for a while, and then they start to pop out those smaller multicellular propagules. Here's another multicellular strand from the other experimental population, B5. They also stay together for a while, and then they start to pop out single motile cells, and then they start to form those multicellular clusters again. So there are these two, the multicellular strands from those two populations differ in their life cycles, but they both form those structures by the failure of daughter cells to separate upon cell division. And most importantly, they form those structures even when the predators are not around anymore, meaning that there's a genetic basis underlying those morphological changes. So to find out what kind of mutations happened in those evolved algae strands, we did Illumina whole genome sequencing. So for the two experimental population and one control population, we pick up multiple colonies to do whole genome sequencing. We also sequence the two ancestral strands to know what genetic variation existed in the beginning, so that we can know what actually happened in those evolved algae. So what we found out is that it appears that it's two different genetic bases underlying two origins of multicellularity in your experiment. So here are the mutations occur in each strand. Each row is an evolved isolate. The first four are from the B2 population that pop out multicellular propagules. The next four are from the B5 population that pop out single-cell propagules. And the last four are from control population that remain unicellular. And each bar is a mutation happened in their genomes. And you can see that for the multicellular isolate in the B2 and B5 populations, they share a certain set of mutation altogether, suggesting that those are, or at least some of them are important for those multicellular phenotypes. So how do we know that which mutations are actually responsible for those morphological changes? We did these analyses called bulk segment analysis. The idea is that, let's say there are 3 mutations in our evolved isolate, and we want to know whether it's A, B, C, or A, B, B, C, A, C, or A, B, C together that are responsible for the phenotype. You can mate it with a wild-type strand to generate a group of cells with different combinations of those mutations. And then we can select for the phenotype we are interested in, which is a multicellular phenotype, and then those genotypes associated with the phenotype will be enriched this way. So to actually do this with algae, we mated our evolved multicellular phenotypes with a unicellular strand to generate an F2 population with different combination of the mutations. So some of the cells are multicellular and some of them are unicellular. And then we select the multicellular phenotype by centrifugation briefly, so that the bigger one should sink to the bottom more quickly. And then so that the bottom ones go to the multicellular pool and the cells on top go to the single cell pool. By repeating this for multiple times, we can enrich the multicellular genotypes and unicellular multicellular genotypes. And then we sequence these two pools of cells to see the allele frequency differences across their whole genomes. And it turns out there are multiple regions in those algae's genome responsible for the multicellularity involved in our lab. So here's the results from the multicellular isolates from the B2 population. The y-axis means that given the overall allele frequency in both unicellular and multicellular pool, these allele, the chance these allele appear in the multicellular pool, and the higher the dots are, the lower the chances are, and the more likely they are associated with the multicellular phenotypes. So you can see several peaks here, and 3 of them also match to the mutations we identified through whole genome sequencing approach. And then there are also a couple other peaks there, suggesting certain genetic background is important for those new mutations to arise. Here's the results from the B5 isolates and the other population that evolve multicellularity. And we also see a similar trend that one of the peaks match to one of the mutations we identified, while there are several other peaks that suggesting it's the ancestral genetic background plus the mutations responsible for those phenotypes. Both these mutations and the one I showed in the last slide, none of them, they are either in introns or intergenic regions, so none of them change amino acid sequences. So we're still working on what they actually do to change those phenotypes. To summarize, we evolved multicellularity in the lab with predators. We have identified mutations that happened in those evolved algae. It appears that it's 2 different genetic bases underlying those 2 different origins of multicellularity. And we are working on the functional relevance of those mutations at the moment. For future directions, now that we have evolved those simple multicellular structures, we want to know how cellular differentiation would evolve. And another postdoc in our lab is working on this project right now. She is presenting a poster today. If you haven't talked to her, you should. So what I presented today is multiple people's work, especially Matt, who oversees this whole project. And we would also like to thank our funding sources. Thank you.
We have time for some questions?
Some questions?
Yes.
Oh, whoa. Nice. Oh.
It's not a basketball player.
Neither did I.
Great talk. Thank you much. I know you're working on elucidating the genetic basis for the drive towards multicellularity under these circumstances, but do you have any sort of predictions or hypotheses about what the phenotypic, results of these genetic mutations are? What would drive the the collective nature?
I mean the phenotypic effect of the. I mean, we were guessing probably something involving cell cycle that usually when cell goes through division, they will form those 2, 4, or 8 cell clusters. But then those mutations might affect this process so that they couldn't separate after that.
Great, thank you.
But they don't look like it's in the known genes we know that's involved in this process.
Okay,
yes.
Cool.
Other questions? Okay. Oh, yes.
I have a question. Green algae are bicons, and all multicellular organisms are unicons. So is there any way that you can try to do any studies on some single cellular unicorns and try to see if you can do sort of explore the origin of multicellularity in unicorns.
What do you mean by unicorns?
It's a two major clades of eukaryotic
Okay.
species.
I know it's possible.
Okay.
Yeah.
Can we thank our speaker one more time, please?